So our next speaker is Eva Balog. She's an associate professor of chemistry in the College of Arts and Sciences at the University of New England. Eva received her PhD in molecular cell and developmental biology from the University of California, Santa Cruz. Banana slugs?
Go banana
slugs. And her BS in biology from the California Institute of Technology. And if I have it right, you grew up in BZ? So went to the West Coast, took all the knowledge, came back home. Good job. All right.
Boomerang.
She also trained as a postdoctoral fellow at Los Alamos National Labs Center for Integrated Nanotechnologies before returning to her home state of Maine to join UNE in 2014. Her research uses biochemical and biophysical techniques to create and characterize novel proteins for various applications in biotechnologies and biomaterials. One really relatively recent, you're looking at me because you have no idea what I'm going to say, right?
I've totally forgotten
All right,
whatever
so
I
one
gave
relatively
you, yeah.
recent reason Eva was in the news is that she is one of 4 faculty members at UNE who has received a recently awarded 5-year, $10.8 million grant from the National Institutes of Health to support an institutional development award center of biomedical research excellence for a UNE Center for Cell Signaling Research. This is awesome. I'm sure she will be happy to describe all the stuff that they're going to do with that once it's up and running. So,
yeah.
Awesome. And Eva's pop culture choice is The City and the City by China Miéville?
Miéville, I think.
All right, neither one of us know, so we'll
It's a good read, though. Okay. Thank you to Kate and the organizers and sponsors of the Maine Science Fest for giving me the opportunity to talk to you tonight about something I love, which is making proteins and making things out of proteins. So I'm going to tell you why anyone would want to make things out of proteins, how we make proteins in the lab, and some of the things we've been doing recently with the proteins that we make. So why build things out of proteins? Nature has shown us what can be done with these building blocks of life. Proteins can, is there a pointer? There is a pointer. Proteins can spontaneously self-assemble into complex geometries like this viral capsid, and they do so due to just inherent energetics in their geometry. And for the same reasons, proteins also excel at specific molecular recognition, as when an antibody is part of our immune system recognizes such viruses. Protein materials can have extraordinary mechanical properties, like this rope-like triple helix structure of collagen, or the silk fibroin protein that, when processed through the spinnerets of a spider, can form threads that have tensile strength that rivals high-grade alloy steel. And it's also easy to forget from these static images that proteins move as part of their function, and their dynamics are intrinsically linked with their functions, as hemoglobin here toggles between 2 shapes, one that has high binding of oxygen and another that helps release oxygen and allows hemoglobin to transport. So proteins do amazing things. I realized I didn't even talk about enzymes, so put that somewhere too, but they do amazing things. Many of the proteins we've made recently in the lab are derived from another connective tissue protein called elastin, which, as its name suggests, confers elasticity and resilience to our tissues. And when you dissect out the portions of the elastin protein that are responsible for that elasticity, they also have another very cool function, which is that they are responsive to external stimuli, by which I mean they undergo this reversible and tunable transition that is showing here slightly sped up in the video from soluble, where it's a clear solution, if you could run it again, soluble, clear solution to, they form these phase-separated particles when you heat them up, but you can also use salt or pH or the presence of certain molecules as your external stimulus and get this reversible switch-like transition. And this is this high tunability of these materials as smart polymers, but smart biopolymers, makes them appealing ingredients for all kinds of applications, from drug delivery vehicles to tissue engineering scaffolds to actuators and soft robotics. So many proteins beyond elastin have compelling functions that we might want to integrate into technologies and advanced materials for the future, but they've had trouble crossing over as well as things like silk and collagen have. Some of those barriers are economic and process and engineering problems, but some of those barriers are biochemical and biophysical challenges, and that's what my lab tries to address. How do we produce the protein? How do we keep it happy after we make it? How do we tweak it so that it works in different contexts? That's what we're doing. How do we make the proteins? You may be familiar with the central dogma of molecular biology, DNA to RNA to protein. Well, the amino acid sequence of the protein is what gives it its function, and the genetic code is what codes for that amino acid sequence. So instead of making proteins one block at a time, we use engineered E. coli bacteria as little factories, and we give them the instructions in the form of DNA. So we grow the bacteria in these sciencey looking flasks, we harvest the cells, we say thank you to the bacteria, and then we burst them open, we take the protein, we purify it away from all the other bacterial goo, and we characterize it with instruments like the calorimeter, which can measure the small amounts of heat changes that occur when one protein touches another. So very quickly, 2 projects that are going on in the lab right now. One of them is incorporating these stimuli-responsive smart polymers into electrochemical sensors to measure quality control for advanced cell and tissue biomanufacturing. And another as part of the Center for Cell Signaling Research, and I want to talk to you about kidneys as well, but is making materials for the study and control of poorly understood signaling pathways that are involved in the cell-cell communication in wound healing, and also beyond. So the question that I pose to you, and I pose myself every day, is what kind of future do we want to write using the protein alphabet? Thank you. Yes. I mentioned electrochemistry, yes.
So how do you mediate the electrons from the protein?
Yeah, so what we're doing is impedance-based non-Faradaic electrochemistry, since you asked. So it's actually, you have to have some sort of charge carriers in the solution, and we're looking at the change in impedance, which is like resistance but a little fancier, as the protein changes conformation, changes shape on the surface.
So it's not a formal electron charge?
Nope. We're hoping to do it without redox at all. Yeah, as opposed to, say, a glucose sensor, glucose oxidase sensor, yeah.
I get the idea of having it, as be the base for something else, like when you're growing new tissues and things like that. What, from a standpoint of people sitting in this room, how would it be useful, or how would it help people that were sitting in this room today?
How the proteins in general,
how
or in
in
the case of
your research in general?
Oh yeah. I would like new cartilage. Someday I might want a new kidney or a new pancreas or cells that could replace that. Mm-hmm. And actually, what Anastasia was talking about, the blood vessel problem has limited a lot of the promise of stem cell therapeutics. Mm-hmm. If we could understand how to create environments that would help cells, that direct them and control their differentiation and their architectures as well as our own bodies can, and then interface with our own bodies once we put those materials, take the stem cell, put it in a nice little environment, then put that into the patient and have it connect and anastomose with all the blood vessels and do exactly what our body could do but better. We need to understand all of the language of cell signaling in order to make that reality. That's the hope.
Nice.
What are you most excited to spend your $10.8 million on?
Well, first of all, it's not my $10.8 million. The lead PI of the project, Derek Molliver, is, he got the money. But there are 4 PIs. I'm one of the 4 project leaders. And all of us have research related to somehow cell signaling, but more specifically aging, inflammation, metabolism, and pain, which are all very closely related. So I'm really excited about the collaborations. I'm also really excited to take my first PhD student through the UMaine Graduate School of Biomedical Science and Engineering. That's one of the things that this is enabling, and I'm going to have somebody starting this summer. So, yeah.
I'm sorry. But, and I guess maybe for all of you too, like how close are we? I'm 55 years old. how close are we to maybe being able to find something that reverses Alzheimer's, dementia, and stuff like that by all of your research? Because that's just an awful, awful disease that seems like everybody's getting nowadays.
Yeah. Gosh, I wish I knew. I think we're. I think.
I'm not going to hold you to it.
You know, I think people are realizing that the individual disciplines are much more connected than we're trained, right? So I was a structural biologist and I studied cell signaling in the context of cancer for my PhD, and I didn't think about metabolism until I had to teach it, years later. And then I realized that the same receptor that's controlling cell division is also making choices about fatty acid metabolism, and I wasn't taught that way, right? So I hope that, and that's just within biochemistry. So I do think the interdisciplinary conversations are going to accelerate things a lot, as well as whatever promises get delivered on AI. But yeah, I don't know, and I hope it's soon.
Thank you, Eva.
Thank you.